Hello, Singapland. My name is Michael Corellia, and today is Friday, April 25th. This is episode 170 of SingApp 10, the SingApp 1 podcast. Tomorrow is Sprint for SingApp, and I want to thank everybody who's hosting an event, especially the Tavillas who are hosting a big event in Boston, and Zoe who's hosting one in California, and everyone else who's hosting one everywhere else. At this moment, we have raised $194,000 via Sprint for SingApp. Of course, a non-trivial portion of that always comes from the Tevilla family, which is just amazing. But 194 is where we're at right now. Hopefully, we'll close higher than that. Maybe 250, 300, 350, who knows? But raising these funds is essential for us to do the work we are doing, and it is nonstop, guys. I can tell you right now, I have had more fascinating conversations with scientists and lawyers and Catherine this week than ever before. The intensity and the pace of our work is furious. And so please know we are working like crazy to make sure these donations benefit our children and improve their future every single day. For my part this week, I also flew to Boston to speak on a panel at the World Orphan Drug Congress. Very, very cool conference. A lot of rare disease people and scientists and biotechs and all the things there. Saw a bunch of friends, saw Hebbian Bio, Unravel, Unravel, who's an independent consultant who spoke at our meeting, Balaji, who's looking at drug combinations, and then I also saw some SingApp families, Heather from our board, Anthony, one of our directors, and Encarnacion from Spain was there. She is a mother of Celia, who I think is 20-something. I'm not going to put a number on it. I'm just going to say 20-something, and that was incredible to see her. She's been fighting the fight for decades. And it was just lovely to have so many SingApp families all together. I also ran into Rachel Heilman, Dr. Rachel Heilman, who gave the drug talk at our conference. And Rachel continues to be an amazing advisor to SRF, helping on all things repurposing and pharmacy. If you missed Rachel's talk at our meeting, I shared a link in the show notes. Make sure you watch it. She's really exceptional, and that was a great talk. Somebody reminded me we had just put it up in the playlist of all the Family Day talks. By the way, all the Family Day talks are up. There's a playlist, links in the show notes. But I wanted to I saw Rachel's, and I was like, oh, it was such a good talk. And I started watching it for a couple minutes to make, just to spot check it. I watched the whole thing. And it was me and Rachel talking, but I just couldn't stop watching because she's so good and so wise. So there's a lot of knowledge in there. If you give your Singappian drugs, that is a talk you want to make sure you've listened to at least once a year. And the other thing that was super interesting on this panel was we were talking about how to pick an indication, right? So if you're a drug company and you have a drug, like which indication should you go after? For example, SingApp 1, obviously, that was what I was pushing. And we were talking about like a lot of small molecules, like think NAL, think longboard, think basically anyone with a regular old epilepsy drug is coming to market, and these are small molecules that apply broadly. And they may benefit one population like SingApp more than another, but it's one product that affects a broad number of diseases, and there's on-target and off-target effects. And then we have what we call precision medicine, where it goes in and it fixes the root cause of the disease. And this would be the ASOs that upregulate SingApp expression, right? And that is just like a narrow thing. And Anna Mignarance talked about, this is her idea, not mine, talked about the T, like the broad small molecules, and then the line down would be the ASOs for the precision therapies. And I was thinking about that, and we're like, is there anything at the bottom of that? Is there anything that's broad and precise? And we were like, no, that can't be. And we're like, actually, no, we're wrong. Tevard. Again, Anna thought of it. I didn't. Sorry, Daniel. And that's cool if you think about it, because what Tevard does is it can correct a specific mutation, right? Like if you have an arginine going to a stop, or if you have an R and an asterisk in your P address, then they have a therapy that fixes that. And then if you have a Q stop, that also, anyway, this is details. And they can fix that for SingApp, for TNNB1, for whatever, right? If you've got a mutation at that level, it is a broad but precise therapy. Super interesting conversation, and it got me thinking more about ASOs. And then I turned around and I got an email from a mom who had heard me talking about ASOs, and her son is missensed, and she's like, so am I correct? And actually her email was very good. I'm just going to read it to you guys because it was excellent. So she's talking about her son, who is a missense mutation. And she says, I have a question regarding ASO therapy. As I understand it, in the case of missense variant, both gene copies still produce protein. Correct. But it remains, well, we think it's correct. We don't know. Because remember, every single missense is different. So until you study them independently, we don't know. But it remains unclear what exactly is going wrong in terms of functionality. Yes. Since ASO therapy increases protein production from the healthy copy of the gene, it seems it may not be effective for missense variants. Could ASO therapy still be helpful for a child with a missense mutation? Very good question. Short answer, we don't know. And I don't want to confuse the answer with the source of the question, which was trials. So let's talk about just missense variants right now. Every single missense variant is misfolded differently, and therefore we need to study them independently. This is why we have the grants in to the people in Finland. Thank you for raising money, missense families. And this is why we have built the SynGAP missense server that is being hosted at Finland right now, where researchers can throw up 3D models of these genes and study the mutations in many of the cases. We still don't know, though, until we put it in a dish or a model system like an animal or a frog or a cell line or whatever and test it out. And I just had a meeting 2 days ago with somebody who's got yet another way we can do that. So we are really thinking about this. But the current state of missense variants is we do not have a way to definitively say for each and every one, this one is not a big deal. This one is a big deal and we shouldn't make more of it. And this one is functionally a null. This one is, you're basically protein truncating. We don't have the ability to say that right now. End of thought. Now, for trials, for clinical trials, what we need to remember is that the clinical trial is where you go in and you use an experimental novel medicine in humans to show that the drug works. So in our case, we need to show that this drug goes into humans and ameliorates the symptoms they have from SYNGAP1 and increases SYNGAP protein expression. And then once it is approved, there can be other trials looking at other populations and uses. But more importantly, once you have marketing approval, once it's sold on the market, you no longer have to go to the FDA. You can say, hey, my kid's got SYNGAP. Here's an ASO for SYNGAP. Doc, will you write this drug? And then you got to deal with the payers. But once it's on the market, it's a lot easier for us to make it available to other populations. However, in a clinical trial, you are looking for the cleanest start point and the clearest signal that it works. And so any of us, if we were starting a clinical trial, would look for the most consistent and homogeneous population to start with. And for SYNGAP, and then depending on the biology and all this stuff, they might want young kids or old kids, or they might start at age 2 or age 5, or they might go to adults, or they might stop at 11 because they don't want to do puberty. Who knows? Who knows? That'll be drug specific. But for pretty much any SYNGAP trial for a precision therapy, such as an ASO, it is my expectation that we will be looking for protein truncating variants. So that's nonsense, frame shift, intronic that has been shown to be PTVs, any of those. Protein truncating variants, excluding the first 3 or 4 exons. Right? And I haven't even talked about the first 3 or 4 exons. We have a blog out there where as soon as we published it, we started editing it. It's going to change a little bit. I'll talk about that soon. But you know who you are, 333 Dells, right? So basically, my expectation is that in a clinical trial, they will want the most consistent, homogeneous population, which will be after exon 4 and protein truncating variants. So yes, missenses would be excluded. And in that time, we should be working furiously to determine which missenses are essentially protein truncating, because then they should get the ASO. But it's not a question of can you get the ASO or not, missense people. It's a question of do you want the ASO? What we need to do is figure out, is that missense variant doing nothing? In which case, yeah, let's make more of it, because then we'll make more of the good one and we'll be good. Or is it doing something bad? In which case, we don't want to make more of it, right? This is not a I want access to the drug question. This is a let's understand this disease question so we can help our kids the best way possible. So just some thoughts on ASOs, which hopefully are not too far away. No matter what, though, trials are coming. So if you are not diligently taking notes on your kid's presentation on the drugs your kids is taking, on the way your kid is presenting. If you don't have your phone full of videos of your kids showing different things, and you don't have a sense of what meds, what supplements, what other changes affect your child, you're not really getting ready for trials. Because when trials come, you're going to be asked to make a lot of observations. And I would just urge everybody to sharpen the pencil and start keeping regular track of your kid now. Because when it's trial time, that is a muscle you're going to want to have built. All right, let me keep up with news. We finally put out this roadmap on our press release. This press release on our roadmap. So we talked about a roadmap, and if you're on our physical mailing list, you should have gotten a printout of the roadmap. Beautiful paper. I hope you all read it and shared it with people. We just finally unstuck, and by we I mean I unstuck the press release, and I want to call that out. Links in the show notes to LinkedIn, because I want to thank. There were 3 or 4 people not with SRF who said nice things about SRF and praised that paper in the press release. And that was really kind. And it's just a great endorsement when your peers are willing to go on the record saying we did a good job. So thank you to them. Other quick news updates. We have a new Warrior story up. Oliver's Warrior story is great. Check it out. If you haven't done a Warrior story yet, if you're a newer family, please do a Warrior story. It is so helpful for families and industry partners to read about our kids and see what's going on. And as you get more and more involved, you can just add a link to your kid's Warrior story from your signature, and people sort of get to know you and your kid. The conference is 222 days away. That sounds like a long time, but it's not. Pre-register now. Book those dates in your calendar, December 4th and 5th. Every conference is a must go to, but as we get closer and closer to trials, guys, if you're on the fence, don't be on the fence now. This is a meeting you want to go to. And that's in 222 days. The impact report, I would say, I want to know also, we have released it. It is on our website. Virginia and I did a webinar, and the Crack Communications team took that webinar and broke it up into like a little mini video for each slide in the impact report. Amazing work, amazing work. So if any of you are presenting about SRF or sharing the impact report and you have questions, you can go to those little videos. You can hear Virginia and I talking about each page individually. Truly excellent work. I'm so excited, Ed. And then I want to flag, one of my heroes wrote about another of my heroes, really good work. And this is Effie Parks of Once Upon a Gene, writing about Terry Jo Bichelle, the founder of Combined Brain. And Effie just decided to write an article to the world about how amazing Terry Jo is. And I couldn't agree with her more about everything, which is why I'm sharing it. And I share the link to that post in the show notes. Please read that and understand how special and important Combined Brain is. Not because, you're ever going to meet Terry Jo Bichelle or you'll have a chance to thank her in person, which you should, by the way, but because I want you to understand how powerful Combined Brain is, and that SYNGAP Research Fund is a part of it. Indeed, we're one of the founding members of it. I've been involved in Combined Brain since day one. And read that article. Terry Jo is really special, and she's building something really new and powerful with Combined Brain, and we are a part of it. In fact, one of the things that's most special about it is through Combined Brain, we have a biobank. A biobank is where we donate blood and plasma and all the things, and those things can be available to researchers when they need them. So this is when I talk about the blood drive. There's collection happening in different states throughout the year. There's a link in the show notes. Read the blog. But this weekend, it is happening in Tennessee. So if you are near Tennessee with a Syngapian and family members, and you would be willing to go and donate blood, please do that. I want to also mention that this biobank can hold more than blood, right? We can also hold tissue. So if your Syngapian is undergoing an operation, or God forbid, if a Syngapian dies, we have the ability to capture, if they're having a biopsy on their gut, we want some of those gut cells, right? If they're having a VNS, we want some of that. We want some of that tissue. If you're doing a intrathecal puncture, we want some of that cerebrospinal fluid, CSF. Like whatever you're doing to Syngapian, if we're cutting them open, we want a little bit of that tissue. Because that is invaluable to researchers, guys. So, and we have all that under IRB, big official medical stuff, everyone involved, top shelf, best practice, all because of Combined Brain. So I just want to end on that note. Go back to your Sprint for SYNGAP preparations, raising money for SRF. We are putting it to good use. I can't thank everyone enough. SRF is moving at light speed right now. It used to be I told you everything was going on in one episode of SYNGAP 10. Like now, I have a list of things that I can't talk about right now because they're mid-flight. Like whatever I say now will be different in 2 days, it will be different in 5 days. And as soon as these planes land, I'll be giving you guys more good news. But there's a lot going on, and if you're fighting the fight that is raising and taking care of us in Gappian, just know that we are doing our best, and help is on the way, and we are grateful for your work to keep these Syngapians going. Keep it up. And if you can raise money and donate to the biobank and volunteer with SRF, all the better. Thanks so much, guys. Talk to you soon.